Good. So today we are going to review two chapters. Very important. I know it's a lot of information, but because this class is online, and also in the week of um, Thanksgiving, uh, we are missing that Wednesday. Um, I need to pack these chapters in this way. Okay. So today we are going to review chapter 12 about classifying eukaryotes. We're going to see that it's a very diverse group, very important, but also viruses. Okay. Next class, next Wednesday, November 17. It's very, very important. Okay. Don't miss it. Okay. This is one of the classes that is the core for microbiology. And for me, is the reason why you are taking this class, okay? So remember that Exam 4 is happening after having these three chapters between November 18 to 21, okay? So this chapter 12, classifying eukaryotes, we are going to see protozoa, fungi, algae, and molds, and actually some about the insects, other important eukaryotes for microbiology. So, as I mentioned, it's a very diverse groups. Remember that as eukaryotes, all of them have a nucleus, right? That this is something that you don't see in bacteria because bacteria are very small. So all the DNA, the ribosomes are spread in the whole cytoplasm. But eukaryotes are larger and have organelles, right? This is right there under the microscope, a huge difference. Other important difference is eukaryotes can reproduce, means that, similar to bacteria, there is also a sexual division, binary fission, body fragmentation, spore formation, but having the gametes, similar like in humans, um, cells that are having half of the genetic material that comes from the germline that stands for female, and another germline that stands for male that also have half of the chromosomes, increase the level of diversity, okay? So having gametes, it's the key to have sexual reproduction. In many of these could be internal, external, could be self-fertilization, or like happened in different organisms according to their stage in the life cycle, could be asexual and sexual at the same, in the same organism. So when we are talking about nuclear division, it's the regular mitosis. When the cell is committed to divide, the genetic material is replicated. And remember, at the end, we end up with two daughter cells, right, that have two nucleus, right, but only in mitosis ends when you have the cytokinesis, that you have that these cells totally divide, right? Having a ring here that totally separate the two new daughter cells. But in meiosis, this is a little bit different because in meiosis, it's separated in two phases. And what is more important, this might be an exam, so be aware of this, that in prophase 1, you have recombination of the chromosomes, right? The ones that you inherit, for example, in humans, the ones that you inherit from your mom, the ones that you inherit from your dad. Now they are switching pieces, large pieces of the chromosomes, right, to create your own blend in the cells that are going to give origin to eggs in females or sperms in males, right? The same thing happened in all eukaryotes. Doesn't matter that is one cell organism or multicellular organisms, doesn't matter. All meiosis processes in prophase do recombination, the crossing over of the chromosomes, switching large pieces of these chromosomes, and as a result, at the end of meiosis 2, you have 4 daughter cells, right, with half of the genetic material, but all of them having different genetic information. And this is why you see, for example, in humans, that if you have several siblings, all of you are different, right? Because all of you receives different genetic information from your parents, okay? Do you have questions? No? Okay. So cytokinesis in eukaryotes is something that, you know, could be all the way when you have total separation of the daughter cells, or might not happen. This is the kind of things that we see in fungi. Or this process that is very related to protozoa, only to protozoa, and it's called schizogony. And here you have further division of the nucleus, right, that you are seeing here. The mitosis keeps going on and on, right, having several nuclei, but only at the end is when you have cytokinesis, right? So as you can see here, you have multiple mitosis happening, but only at the end you have the 8 daughter cells. But all of them have, um, you know, 2 sets of the chromosomes. So in this case are not haploid, they are diploid, the complete sets of chromosomes. And as you're going to see next, some of the classification of these eukaryotes at the beginning, late 1800th century, was mostly based in how these organisms looks. But through the research and time, we realized that this is, not really reliable information, because during evolution, you have animals or microorganisms that are very much alike, but actually they share a very different evolution. So this is why today, in the actual classification that we use today, we have fungi separated from plants. We have protozoa and different groups, algae also separated from plants, and animal, that is pretty much his own group on their own. But as you can see here, and this is what we are going to see next, eukaryotes are a very diverse groups. Most of these are unicellular, okay? So don't get confused because they are just one cell big. Here, the thing is, they are eukaryotes, okay? They have nucleus, they have organelles, and this is why they are eukaryotes, okay? So looking at the protozoa, most, they are unicellular, very diverse in shapes, and they don't have cell walls because this is something of bacteria, right? Some of them are motile, using cilia, flagella, or pseudopods. And in nature, most of them are free living. Very few are pathogens. And this is what we are going to focus on the next slides, okay? As you can see here, very diverse in shapes. They may have cilia like paramecium or dinidium, or may have flagella like trypanosoma, euglena, or pseudopods like amoeba. Most of them have just one nucleus, but this is biology. So you may find organisms that looks like joint twins that you have two nucleus, like Giardia. Mitochondria, remember, is these very important organelles that process energy for the organism, right? But here you will find some protozoa that have no mitochondria, but something that resembles, in terms that is called the kinetoplast, have a similar function or may have several, I mean, hundreds, okay? Some of them have contractive vacuoles because this is how they manage water, but also help them to work some kind of a stomach, okay? About the nutrition, most of them are chemoheterotrophs, means that they eat bacteria or other microorganisms or from the host, right, to have their energy, right? Very few are photoautotropic, right? Means that they can do photosynthesis. Most of the protozoa reproduce using asexually methods like binary fission or schizogony. Some are sexual. Remember, they make gametes that we call male and female, right? And it's very common to see alternation of generation. So I'm going to explain this using one life cycle so you can see how weird that looks for humans, okay? So different stages in life cycle. So we have the, for example here, the trophozoid and the cyst, okay? These trophozoites is usually motile or the type of stage that get nutrition from the environment, okay? The cyst is usually very resistant to the conditions in the environment. So cyst is a similar situation when in bacteria, we were talking about the endospores, okay? Because this cyst have walls very strong that, for example, in some of the coccidias, you can use the strongest acids that you can think about and they are still good, okay? So the only way to kill them is using UV light, okay? So some of these, for example, like Cryptosporidium or Isospora, have that condition that are very resistant. And therefore, if you have to put in place methods, of the place that you work, the hospital, you need to work hard with bleach, with acids, because it's very hard to get rid of this cyst. Remember, cyst comes from protozoa, okay? Endospores are bacteria, okay? But they have the same mean in the environment, protect the organism from very harsh environments, okay? Questions about this? No? Okay. I know that here you are going to hear a lot of, um, life stages and cysts and trophozoites. So a lot of new words. And it's kind of, hey, why do I need to know this? Well, it's not only because it's part of the class and, might be part of the exam, but I always try to connect this for the kind of situation that you are going to face in real life, okay? So this is why I mentioned in the previous slide about controlling and preventing disease. Because most of the time for you guys working in healthcare settings is going to be your task. You are going to be in charge of, hey, we have a bad situation with cryptosporidium, right? That, for example, is a big deal for patients with HIV. We have to get rid of this. So you have to find very strong conditions to kill this cyst. Okay, good. So, importance of the life cycles. Okay, this is something that changes according to the organism. Okay, sometimes they have similar patterns, sometimes changes, but this is what is more important. Help you to understand the vulnerable points during the life cycle when it's the best option, to find treatment or prevent, the transmission of the disease. So let's take a look at the classification of protozoa. Okay, so far, this is something that might change over the years, but so far it hasn't changed in the last 10 years. We have 6 different groups. Okay, most of this is based on DNA sequences. So the first group is called Parabasala and is related to this formation that you are seeing here. Okay, this parabasal body. This is unique to this group. Half flagella, as you can see here, and undulating membrane. Remember, this is unicellular organisms, but is not a bacteria. Why? Because you are seeing a nucleus, okay, right here. One organism that is very important for humans from this group is Trichomonas, okay? Trichomonas can live in acidic environments, and it's especially important because it's a sexual transmitted disease, okay? Creates vaginal itching, burning, full smelling, vaginal discharge, okay? And the organisms use this axostyle, to anchor to the tissue, okay? So Trichomonas vaginalis is one that is very common, okay? Another group is called Diplomonadida, okay? And here we have this very interesting organism called Giardia lambia. Giardia, the cysts of Giardia are common in water sources, okay? This is one of the reasons that water are treated with, different chemicals, ozone, chlorine, to kill microorganisms. Because if you have the infection by Giardia, right, you ingest the cyst. Remember, the life stage that is good to handle all this situation in water, under the sun. But once that cyst is ingested, you have the trophozoite. That is what you are seeing here. These have no Golgi of peroxisomes, but have 4 flagella and 2 nuclei, okay? In a future chapter, we will see that Giardia have feeding disc. So right here, have a system that creates like a vortex and damage the small intestinal cells, the epithelial cells. So in the kids, but also in adults, but more frequently in kids, by damaging the epithelial cells in the small intestine, creates a pathology that is called malabsorption, okay? Means that you are heading into malnutrition, not because you are ingesting few food or poor nutritional food. Is more related to your inability to transport that nutrient from the lumen of the gut into your tissues. Why? Because the cells are being destroyed, okay? So the villi is pretty much gone. Your intestine looks pretty much flat, okay? Amoebozoa. This group has been going back and forth, through time. Some people consider this in one group, and now it's a protozoa. So have pseudopods, no shells, sometimes are called slime molds. So this is another reason why it has been moving around in the taxonomy. But here we have 2 important groups for humans. Acanthamoeba that is also a waterborne disease that, if the water is not correctly treated, might affect the central nervous system. Okay, it's able to go inside of um, um, move across the bloodstream and colonize your brain. Okay, so it's usually found in the cerebrospinal fluid. When the doctor samples cerebrospinal fluid, it's found there. Okay, it's very hard to grow, very hard to culture. Another important species is entamoeba, and this is another emerging STD, right? And usually damage the colon because colonize the epithelial cells and break them. Okay. Another group, Euglenozoa, this one have something that resembles, mitochondria. But what is important here is two major groups in Kinetoplastida, Trypanosoma and Leishmania. These two are not big deal in general in the States, but here in Texas are important because trypanosoma has been found especially in dogs, but from Central America, South America might be common. Okay, so trypanosoma have this very interesting shape that you are seeing here with a flagella that creates undulated membrane along of the body. And some of these trypanosomas can be free living in the blood. In Africa, it's different because the trypanosomas in Africa lives inside of the red blood cells. Okay, so it's the same group, but they choose to live in different areas. Okay, and these two, according to the species, mostly Trypanosoma cruzi, can give you mega viscera. Okay, so colonize your colon tissue, your spleen, your liver, and expand the size of the tissue in a way that is 10 times larger what it's supposed to. Cruzi also can colonize your heart and affect the cardiac rhythm. The other one that you are seeing here, Leishmania, is very common from Mexico all the way down to Venezuela, Ecuador, Brazil. And this one gives ulcers, like what you are seeing here. Okay, this is an intracellular obligate parasite. Okay, and what you are seeing there is a life stage called a mastigote. Okay, the treatment for this is quite painful because it's a solution that is very thick, looks like oil. Okay, and it's dosed for 21 days, so it's a long treatment. Okay, another group, alveolates, and here have different groups. Here you have ciliates, apicomplexes, and dinoflagellates. So in ciliates, we have Valentinium, is the only one that is known pathogenic to humans. Most of them lives in water, usually just give you diarrhea. Apicomplexes, this is more important for humans. Here we have Plasmodium, that is the origin of malaria, affects millions of people, and we are going to talk about this in a different chapter. Cryptosporidium, very common in people with HIV, Neospora and Toxoplasma. These two are more common in animals, but Toxoplasma is especially important for pregnant women, okay? Because only from cats you can get this. However, the stage that you are seeing here, that is called the cyst, the tissue cyst, um, can be ingested by eating contaminated food, poorly cooked food from pigs or other animals. Actually, cows can also have toxoplasma, also by blood transfusion, but only the cat is the one that is able to complete the life cycle of toxoplasma. However, adult cats are not the issue. Only kittens are the ones that complete the whole life cycle. So most of the doctors will tell, hey, if you have cats, watch out. But it's only kittens, when they are adults, they are no longer able to transmit the oocyst. That is, what is, like the most infecting thing on toxoplasma. And usually you get infection with the oocyst when you are in direct exposure with the cat feces, okay? So remember, one way to control this is avoiding exposure to the cat feces and cooking the meat thoroughly, right? Okay. Malaria. Malaria, as I mentioned, is a very important disease, affects millions of people worldwide. We have it here in the Americas. Well, it's very common, in the south of United States. But still today, Central America, South America, in Africa, south of Europe, so affects millions of people. And it's transmitted by mosquitoes that can take the sporozoites, right? That stages that are able to create the gametes, right? That once they multiply inside the human, right, they are able to join in the sexual cycle, creating merozoites that are taken by mosquitoes again. And inside of the mosquitoes, right, they develop into the sporozoites. So in humans, according to the place that you are looking, you might see sporozoites in the liver, right? Or this life stage, the escusons, inside of the red blood cells, okay? So pay attention to this because it's one of the typical questions of the exams, okay? And in the certification exams, okay? Dinoflagellates. These ones are also part of this group. They have a flagella. They are not infecting humans. What happened with them is they live in the oceans, large bodies of water, and they under the right conditions, bloom. And for example, this one here, Carenia brevis, is the responsible for the red tie. This is something that you see in the news, not every year, but every some years. And the big thing about this is makes neurotoxins, okay? So this is why they close the areas and says, do not swim, because very easy that through the oral mucosa or respiratory mucosa, you get in contact with the neurotoxins. And right there, you die after 30 minutes of being exposed because pretty much block your muscles from contracting. Rhizaria is the last group. This one, not really important for humans, just because they have weird shapes, like what you are seeing here. Very interesting geometrical shapes, that are very intriguing, but not important for humans. Okay, so that was protozoa. Okay, do you have questions? No? Okay, so now let's take a look in a different group of eukaryotes, fungi, okay? Fungi, we know them as molds, mushrooms, or yeast, okay? They are chemoheterotrophic, means that they fed in other living things, right? These ones have cell walls, mostly made of chitin, okay? This is the same component that we will find on insects. And they don't do photosynthesis because remember, they are chemoheterotrophic. So this might be an exam. Uh, they are more related to animals than plants. And this is something that not too many people realize, okay? Similar to protozoa, very few species are, creating a big deal for humans, okay? Or plants of animals. So in terms of the morphology, they might go from hypha, that is what you are seeing here, with septums, with these divisions, or without divisions. Or like in yeast, you see the buds, right? Yeah, yeast, very important to make bread, right? Or dimorphic, that is the kind of species that can do both hypha and the buds. As I mentioned before, they fed on organic matter, right? But most of that organic matter is the way how it's already dead, okay? So they are not killing other things that make them saprobes, okay? Means that they are decomposers. They feed on dead or decaying organic matter. However, as you are seeing here, some of them trap and kill microscopic nematodes like this worm that you are seeing here, okay? So this formation here is called the hyphal loop. And as you can see here, traps, right? Is trapping here the worm in order to decompose this worm from here, okay? Um, Wait, later, some of these are able to, using the hypha, they are able also to penetrate inside of the cells, okay? And this is how they fed into the living material. Reproduction, this is very complex because they have the asexual spores. And here you will see that they make spores, but this is not sexual reproduction. They have spores that are called positive and negative. They are haploid, but not actually derived from germline cells, okay? So this is why it's kind of unique, okay? Because you have haploid material, right? But it's not like this is the male or female because there is no differences in terms of the type of chromosomes that these haploid cells or ascospores have. It's, mostly in terms of that they are just haploid. And then when they fuse, right, they can keep growing into that stage now eukaryote, but not really fusing the material. Only when they do the eukaryon is when they are recombining this genetic material, right, to go into diploid stage. And from here, they do meiosis, you see, but actually not really clear differences into gametes like other organisms, okay? Classification, there is 4 different groups. The first one is Zygomycota, and here we have one that is very important for humans, Encephalitozoon cuniculi. This is very common on rabbits, okay? And you can get infection by respiratory airways and might give you, a bad sickness in your lungs or kidneys because once these fungi invade your cells, destroy them, okay? Um, Ascomycota, now this is uh, superficial, it's not intracellular fungi, this is extracellular. And here we have different groups, okay? We have Trichophyton rurum that give you, this toy nails appearance, right? Very easy to get rid of this. You just put your feet on um, vinegar for a couple of weeks, 15 minutes, and altering the pH, you kill the fungi, okay? Or, very important for your health, like Penicillium notatum or Penicillium chrysogenum. That is what was found by the Department of Agriculture in this Penicillium rotatum that grow outside of the cantaloupe is what we use today to obtain the penicillin, okay? Because the species that Fleming found out actually gives little penicillin, okay? But the one that was found here on the States, not in England, is the one we actually use today to obtain penicillin, okay? That was found by the Department of Agriculture. So, other important group here, division Basidiomycota, is the typical morphemes. The only rest of this is that they make toxins or, hallucinatory hallucinatory chemicals, you know, make you high. So this is the kind of compounds that you need to be aware of, right? Take care. Okay. The third group of eukaryotes, algae. Algae have different also life stages. Could be from unicellular to colonial or multicellular. They also divide by sexual or sexual reproduction. And here, their risk is when they bloom, okay? When they bloom in large bodies of water, because we have that they make a lot of toxins, okay? So we have amnesic shellfish poisoning, ciguatera fish poisoning, or diuretic shellfish poisoning. All these derives from algae that actually makes toxins, okay? It's not that they infect you, it's that they make toxins. This is so bad that you need to check, NOAA or Texas Parks and Wildlife for the states, because when they bloom, like for example, the golden algae, uh, you know, might be providing, to swim in these areas because it's really dangerous, okay, because of the toxins. Okay, molds. This is another group. Here, it's mostly damaging for, for food. In the case of fishes, or like in the case of the potatoes, you know, like the Phytophthora infestans that was what caused the Irish potato famine, 150 years ago, and that pretty much pushed half of the population of our island at the time, to move around the world. Okay. Other important eukaryotes of microbiological interest. Here we have fleas, Tunga penetrans. This one is able to live under the toenails or in the skin. So it turns very, painful when you walk. This usually happens when you are just using open-toe shoes, right, in nature. So you have this very tiny flea living in your skin. Other organisms like ticks, mites, flies, mosquitoes, or kissing bugs, they are important because they transmit, okay? They transmit viruses or bacteria or protozoa, like we just take a look. And arachnids, in this case, most of them are, just toxic because the saliva damage the tissue. Only on ticks, there are the ones that transmit Rocky Mountain spotted fever. This is a bacteria, remember, rickettsia. And insects, as I mentioned before, like Aedes aegypti or albopictus, both very present around here in the Houston area, are important because they can transmit different kinds of viruses. Okay, so that was chapter 12. So now let me put the slides for chapter 13. Okay. So now chapter 13. So this is viruses, okay? Viruses that now are very much, on trend because of the COVID-19 that we are having today, right? So remember that one very important characteristic of viruses is they are acellular, okay? And this is why we think that they are not deserve to be considered alive because they don't make a cell, they don't have metabolism, they don't have cytoplasmic membrane, cytosol, or organelles, right? And they depend on cells, humans, animals, or plants, or bacteria to replicate. So they are dependent on the host, you see, in order to grow, in order to expand in terms of the population. So this is why they are not considered to be alive, okay? And here at scale that you can use to realize how small are viruses, okay? This picture here shows you a red blood cell that is around 2.5 microns or 2500 nanometers. Next, you are seeing a bacteria, E. coli. And E. coli right here next to viruses looks huge, right? So here you are seeing the size of different viruses just next to a piece of a bacteria. You see, so they are very, very small, usually around to 25 to a couple of hundreds of nanometers, okay? So viruses are very, very small. This is why if you need to destroy viruses, filtration is not good enough, okay? This is why now during the COVID-19, if you are using cloth, face mask made of cloth, that reduce transmission, but is not going to prevent you to get infected. Why? Because the size of the clots is huge, allows a lot of these viruses to cross, okay? So we use face masks to avoid the transmission. Just in case we have it, the face mask is able to trap the large particles of saliva that contains the virus. So most of the virus, if you have it, will be trapped in the mask, right? But it's not going to prevent that you get infected. In order to do that, you need a tested mask, N95, N98, that can do that job, okay? So viruses could be found in two different states. Please be aware of this because might be on the exam. If it is extracellular, it's called the virion, okay? And usually they have an envelope that belongs to the host cell membrane, okay? So when they are living, they grab a piece of the host cell membrane so they can go stealth, right? They can mimic the host cell components. And they have a capsid, like all the viruses, nucleocapsid, and sometimes a matrix, right? That is these proteins that line the space between the envelope and the nucleocapsid. And if it's in intracellular states, now you have here that the envelope and the capsid is removed, right? And just inside of the cell, you have the nucleic acid of the virus, right? That, as we are going to see next, could be in different ways, okay? The genetic material of the virus could be DNA, could be RNA, single strand, double strand, okay? You have it all, okay? What you are seeing here is the genome, the chromosomes of a bacteria, E. coli. And right here, the viral genome, okay, of Bacteriophagous, that is the ones that lives, predates um, bacteria. Remember that the viruses depends of the host, right? And because of that, they are very particular about the kind of cell, right? About the kind of host, the species of the host that they are able to infect, okay? They're very picky about it because they need to establish a specific connection, like for example, what you are seeing here with HIV, right? This capsid protein to make connection with these two markers, CD4, CCR5, in order to be able to attach and then inject the genetic material, right, that will go to the nucleus in order to infect the host cell. Without these two markers, CD4, but mostly CCR5, the virus is unable to infect the cell, okay? So this is why there is a high dependency between the virus and the host, and actually the type of host cell. What happened today with SARS-CoV-2, that is what give you COVID-19, the spike protein is able to make a connection with a protein, with a marker that is very abundant in different type of cells because it's what regulates the water, okay? So this is why SARS-CoV-2 can be found in blood vessels, in um, digestive tissue, digestive tract tissue, in lungs, in different tissues in our bodies, okay? Some viruses are generalist, like West Nile virus, that if you have been following the news, you know that we have West Nile virus here in Houston, right? Every year, every year you found that there is some small but important number of people that might die according to the age, because as we age, we turn more susceptible to viruses. Why? Because the immune system is not as good working against the viruses or making memory against viruses. So this is when the viruses are able to progress and damage, the tissues. So in the case of West Nile, it's able to infect, uh, mosquito tissue. It's able to infect humans. It's able to infect horses. It's able to infect birds, migratory birds. It's able to infect Culex, Aedes, different kinds of hosts, different kinds of vectors. And this is why West Nile can spread so much among the population. But this is the kind of things that we see here seasonally, okay? Usually during summer, around July to September, October, and during winter, disappear. Okay, and here we're going to talk this later in chapter 15. Okay, so capsid morphology. Capsid is the protein part. One, protein, you will find protein in different parts of the body, but it's usually the coat, okay? The capsid is mostly made of protein that protects, surrounds the genetic material, okay? And that capsid is what gives the virus usually that unique shape, okay? It's very important for the virus to attach to the host. The virus that you are seeing here is a bacteriophage, okay? Is the kind of virus that infects bacteria. At some point, These bacteriophages have been used to treat bacterial infections in humans, okay? They are safe. I know, looks ugly, but they are safe to be used in humans because they only infect bacteria, okay? But according to the protein, viruses may have different shapes, like a rod, like this is a tobacco virus, uh, rabies that looks like a bullet, polyhedral, this is influenza, or, all these different types that you are seeing here, you know, that are very diverse. Even a very complex shapes like Ebola, right? That looks more like a nut. Yes, it's a virus. Okay, this is a virus shape, Ebola. Okay, right here. So the envelope, remember that the envelope is outside of the capsid and is taken when the virus is leaving the host cell, is a piece of the host cell that is used by the virus to go stealth. However, some of the proteins might be transferred to the envelope, right, in order to make this particle to be more infectious to other cells. Okay. Usually, if the virus is outside of this envelope, might be more susceptible to be destroyed. Okay, classification of viruses. This is done by a committee that met since 1966 and is usually done using generic material. However, there is a lot of discussion because depends on mostly on the type of nucleic acid, as we are going to see, but also in other characteristics like presence of the envelope, the shape, size, okay? One thing that will be great for you is in viruses, we don't use Latin. Now we use English, okay? So this is something that makes this more easy to remember, okay? So here, the classification of viruses, you see, we have a big group that is pretty much just having DNA as generic material. Another big group that have RNA as generic material. You see, this is right there, the biggest division. But here's just, some important groups, adenoviruses, that give you respiratory infections. And this is one group that is being used to deliver vaccines, okay? Um, um, in RNA, RNA viruses, it's more common, it's more diverse. For example, here we have the flaviviridi, we have hepatitis C, right? But in a different group, picornaviridae, we have hepatitis A. Hepaviridae, we have hepatitis E. You see? So it's not like, oh, hepatitis, it's just one type of virus, it's just one family. Actually, it's a spread in different viruses, okay? So let's take a look in some of this. Remember that here in Houston, or in general in Texas, we have a very nice system. We have this web page from the Department of Health Sciences Services that they give you a good information about what it's more prevalent in the season, okay, that we need to be more watchful about, okay? Here are some of the viruses that might be important, for you to have in mind. At the first class, I told you about Ebola. That is very common, or more, you have focus appearing, disappearing in Africa. This is pretty much endemic from there. But we have it in 2018. We have one patient with Ebola, 3 nurses infected in Dallas. So it's the kind of thing that, you might say, no, this is not from here. This is Africa. doesn't matter. We may have it, okay? So it's the kind of things that is so important to learn about, okay? And I'm going to just present the information about some major viruses here in our area, okay? Measles. Measles, was the kind of thing that we consider eradicated, but is coming back. Why? Because some parents think that it's no longer important to vaccinate kids. So now you have this virus spreading, right? Doesn't matter that we have a vaccine since 1963, right? It's very contagious by respiratory droplets, right? So it's an airborne virus, and humans are their natural host. It's not in other animals, just host. Symptoms: cough, runny nose, red eyes, fever. But this, the rash, is what make you to be alert, right? And this is when you take your kid to the doctor, and using a PCR test, now is diagnosed to be positive for measles. But remember, we have a vaccine available for this, so we shouldn't be seeing this. Mumps, right? Mumps, it's more common on teenagers, right? Again, humans are the natural host. Very, very contagious. Again, incubation takes a lot, around 2 weeks. We have vaccines in 1967, right? And the major symptom is the swelling of the parotid glands right here, right? And remember that we have a vaccine that with just one shot protect you from measles, mumps, rubella, and varicella. Right there. 4 different viruses in one shot, right? Okay, now another viruses. This is more um, um, hard to get because so far has been diagnosed as a group of symptoms, but it's not clear. We know that it's viral in their origin, but it's not clear if it's an enteroviruses or a rhinoviruses, okay? It's called acute flaccid myelitis, and we have it here in Houston, okay? It's more common in kids, and is usually seen between August and October, so it's seasonal. Not sure how this happened. It's believed to be respiratory, okay? But we know that affects the central nervous system. So by affecting the brain, now you have these 2 characteristics, facial weakness or drop, and droopy eyelids. This is so far what we know, okay? But these kids might be affected for life. Very contagious. And of course, Ebola, right? Remember that this is BSL-4, highly, highly contagious. There is no vaccine. There is, the only way to diagnose this is using blood, right? So if you have this kind of situation, you require to be trained to work with these highly contagious pathogens. HIV, that's similar to COVID, SARS-CoV-2 is what give you COVID. HIV is what developed in AIDS. AIDS stands for human immunodeficiency viruses, right? Or HIV. And here you have that the virus lives in your immune cells. And by depleting your immune cells, it's able to destroy pretty much your defenses. And this is why now you are being colonized by different organisms that create opportunistic infections, right? Diagnosis using the blood. Today, we don't have vaccines, but we are plenty of chemicals that can uh, prevent the virus on the different stages of their replication. West Nile virus, as I mentioned, very common in the Houston area, right? Incubation takes between 2 to 14 days. And this virus is a generalist, can live in humans, birds, horses, right? Give you kind of, um, fever. But especially if you are senior, it's very important that you go and be diagnosed to receive the proper treatment, okay? Viral replication depends on the host. There is two ways for this to happen. The lytic replication, right? When you have that the virus gets in and multiply in so large numbers. And this is in terms of minutes of a couple hours that breaks the whole cells and is released. Right? This is in terms of minutes, okay? This is very, very, very fast. Or lysogenic replication means that may happen as we saw before, or the genetic material of the virus integrates with the host genome and it stays there for several generations. And then at some point, start making viral particles again that again go into the lytic cycle, breaking the host cell. Replication, usually for this happen in stages. First is attachment, right? So that allows the virus to inject the genetic material, the genome inside of the cells, or might happen because the virus tricked the cell to be taken, because have some components of cell membrane trick the cell to be taken by endocytosis, and one is being able to escape, the protein capsid and the genetic material. It's integrated with the genome. As I mentioned before, there is different kinds of viruses using RNA, DNA. So if the virus uses DNA as genetic material, here you have the retroviruses that are able to integrate, like adenoviruses or herpesviruses are able to integrate into the host genome. Other viruses, like retroviruses, are able to, or they have messenger RNA or RNA as genetic material. So from here, they need to be retrotranscribed, means that they have enzymes that allow them to take that RNA into DNA. And from DNA, now they can make the proteins, okay? So having these retrotranscription enzymes right there is a molecular target for biomedical research to do medicines that prevents these viruses. This is why we have so many options for HIV. Okay, and to complete the life cycle of the viruses, remember some of these might be latent, because it integrates with the host genome, or escape. That is what you are seeing here. You have the capsid. Some of the viruses, not all of them, they can break the whole cell, or some of them take a piece of the whole cell, right, in order to go stealth into the host environment. Some of these viruses can give cancer to the host. It's around 30% of the viral infection. Different viruses can give you cancer, because of the place that they integrate in the host genome. Remember from AMP that we have proto-oncogenes or repressors of these oncogenes that according to how the virus disrupted sequences might activate the oncogene or damage the repressor of that oncogene, and this is how we end up in cancer. Okay. How to culture these viruses in the lab? We require host cells, right? So according to the type of viruses, we require bacteria as host cells, or we require cell cultures. And this is why cell lines are so important. So what you are seeing here is a bacterial line, means that this plate was cultured with bacteria, and after the bacteria growth, now it's exposed to bacteriophagous, this kind of viruses that lives on bacteria. And all these holes is the evidence that bacteria are dying, and therefore you are seeing the holes. So this is why people that work with viruses says that working with viruses is like working with shadows, because you are just able to see the damage at cellular level that the viruses are able to do. Embryonated eggs is one way to make the viruses to grow, because embryos are very susceptible. They don't have a fully developed immune system. So this is why you hear that embryos are so susceptible or are used, and mostly from chickens, are used to grow viruses, okay? Or cell lines, usually from cancer, are used, what you have here is a complete uninfected um, culture of cells. And what you are seeing here on the right, it's the same cells, but infected with viruses. And as you can see here, a bunch of these cells are dying. So remember, it's not totally clear if viruses are alive, right? Because they don't have cytoplasm, they don't have a metabolism, they require a host cells to replicate, right? And other particles that are not viruses are the virions. Okay, virions only infect plants, so not really important for humans, but for farming is important. And other element are prions. This is really, really dangerous. Okay, some years ago was a big deal, especially in England. They create this mad cow disease because what you have here is a protein, okay? A protein that is able to transform, okay? Is able to transform the other proteins that work normally into the bad protein that it's able to create the disease. So we have this spongiform encephalopathies, right? There is no treatment. The only way here is to incinerate everything, okay? There is no treatment. There is no cure for this. And this is brain tissue here, normal brain tissue. And here how once the prior particle that modify the structure of the protein and is able to modify other normal proteins. Now you start seeing holes because you have, this is what is called spongiform, because now the cells are dying. So you have like these holes, okay? Some of these viruses are under strict control of the government because some of these might be used to create weapons, okay? This is why you might see even in hospitals that how you treat these diseases is under the strict control of government, okay? Good. So now let's do some practice questions, okay? Remember that you can type your question, I'm sorry, your answer in the chat box, okay? So let me share here. Good. Chat. Okay. So first question. the type of nuclear division in which chromatids partition into 4 nuclei such that each nucleus receives half of the amount of the original DNA is called meiosis. Okay, meiosis. Remember that at the end of meiosis, we have 4 daughter cells with half of the genetic material. This is why they are called haploid. Okay, haploid, which term best describes a cell that contains only one single copy of each chromosome. I just mentioned this. Haploid, right? Haploid. What is the name given to the process of cytoplasmic division? And remember, the mitosis is not complete until this process happened. Cytokinesis. Which of the following is true of protozoa? Protozoa. They are similar to bacteria? No, right? They are eukaryotes and they lack of cell wall, okay? Remember, protozoa are eukaryotes, so they have a nucleus, okay? Unicellular fungus is called an yeast, right? Yeast, okay. So this is it here.